Now imagine you are walking down the street minding your own business, and there is active war going on in your stomach. There's active war between this monster and your immune system. Suddenly you start having heartburn. You start burping like crazy. Probably because of what you ate or because of what you ingested 2 weeks ago or a month ago. Pathophysiology of H. pylori infection. Next. So, how does H. pylori cause infection? What is H. pylori? Helicobacter pylori. The bacteria is helical. It's cylindrical in shape. And pylori because when it was initially discovered, it was discovered in biopsy specimens taken from the pyloric, the lower part of the stomach. So how does this bacteria cause infection? The bacteria enters the body orally through contaminated food, contaminated water, and that sort of stuff. You ingest it, it enters the body, colonizes a specific area of the stomach, mostly the lower part of the stomach. So it's cylindrical, flagellated organism that moves, let's say it has tails and it moves in corkscrew motility fashion. And your stomach has layers, right? You have acid production for digestive purposes. Let's say you have layers, 3 layers, just for to make things easy. You have the acid at the top that would interact with food, bacteria, microbes and pathogens and things like that. So acid at the top, you have mucous membrane, underneath that, and then you have cells at the bottom. So these cells secrete mucus, antimicrobial chemicals, and then there are, of course, the acid-producing cells. So why is this bacteria not damaged by the acid like other bacteria? It has a special enzyme, it has a special mechanism. It takes this chemical called urea from the surrounding area, takes it up, breaks it into ammonia and CO2. And from basic chemistry, if you mix acid with base, you neutralize the acid or the base and you produce a neutral compound or chemical. So the acid in the stomach is neutralized by the ammonia that's produced by the bacteria using the urease enzyme. So once it gets down to the cells, it uses special proteins, lipoproteins, and then outer membranes or proteins to tightly bind to the epithelial cells, or even pass the apical surface and bind between cells where there are gap junctions, things that connect the cells, and stay there and cause problems. So this binding this anchoring allows prolonged persistence despite gastric motility and acid production. So once it binds to the cells, it uses special proteins that have disease-causing potentials, or they are virulence factors, right? So the main protein that this bacteria uses is called CAG-A, and in a needle fashion, it binds to the cells and it injects this protein CAG-A into the cells. And once that protein enters the cells, problems happen. And when that happens, the cells change their shape because it alters the actin cytoskeleton, disrupts the cell signaling, it changes the polarity of the cells. Cells have this thing called polarity. On one side, they release, this chemical. On the other side, they release different kind of chemicals. So they lose that polarity. And it also uses special proteins. So let's say these are 2 cells and there are gap junctions that connect 2 cells. It uses special proteins to just chop off something like HTRA proteins, It chops off those proteins, and the cells become elongated, they lose their shape, and can't secrete mucus, can't secrete antimicrobial chemicals, and they proliferate, they multiply, and instead of remaining attached there, they just float away and they multiply. So in addition to all these changes, inflammatory chemicals like inflammatory cytokines like interleukin 8 and other cytokines are released from these cells. And when that happens, these chemicals are attractants, so they attract nearby cells, immune cells, and immune cells come to the area and release more damaging chemicals. And so that's not the only protein now that's causing, Kage is not the only virulence factor that this bacteria uses. There is something called VAC-A toxin. So this protein, unlike Kage, which is directly injected into the cell, this is a toxin which is secreted and it binds to the cell surface, and then is then internalized through endocytosis. Once it's inside the cells, it leads to formation of vacuoles within the cell, disrupting various normal cellular functions, mainly disrupting the cell's membrane potential and leading to cell death through apoptosis. And when this happens, the cells pretty much get filled with vacuoles, or something that looks like a bunch of bubbles, and they undergo apoptosis or death. So basically, the bacteria gets to the area, neutralizes the acid, binds to the cells, injects these transforming proteins into the cells, and then the cells pretty much lose their function, their shape. They actually get transformed into precursor cells to pretty much a malignancy process. The most interesting factor in this case is this CAG-A protein. The protein gets injected into your cells, the whole cell, by the bacteria. And this protein is not even active when it is in the bacteria itself. Once it enters your cell, the whole cell, it gets phosphorylated, gets activated, and it starts doing the damage. So once the protein is activated inside the cells, it leads to activation of proteins such as beta-catenin, NF-kappa B, and these cells are well known for uncontrolled cell proliferation and chronic inflammation. So this protein directly contributes to the oncogenic transformation. So one thing that's important to note is that there are different types of or different strains of H. pylori. Some H. pylori bacteria have these virulence factors, some of them don't. The ones that have like L-type HTRA protein activities, they have mechanisms to separate cells to cause this transformation of cells, lead to gastric cancer, or severe symptoms of gastritis. So in summary, the bacteria enters your body, binds to the cells, injects these virulence factors into the cells, and the cells undergo dramatic structural, even molecular changes. There's a damage to mitochondria leading to DNA changes, and cells also undergo significant structural changes that are precursor to cancer. So chronic inflammation, if the cells, for example, if the cells in the antrum are affected, where hormones such as somatostatin are secreted, which has a negative feedback mechanism, or acid production blockage effect. With no somatostatin, you can have increased production of acid, and eventually leading to, with chronic inflammation, and with immune cells playing their role, chronic inflammation can lead to Damage from the acid and chronic inflammation anywhere eventually can lead to cancer. So, because of its cancer-causing potential, H. pylori is considered or categorized as class 1 human carcinogen by WHO. And H. pylori is the leading cause for gastric cancers. Lifetime risk of developing gastric cancer by patients who get infected with H. pylori is 1 to 3%. That's why getting the right treatment and having resolution of this infection is very important. And atrophic gastritis, intestinal metaplasia, and dysplasia are considered to be pre-neoplastic lesions. So untreated, uncontrolled infection and cellular changes with H. pylori infection could potentially lead to gastric cancer. Therefore, treatment is very, very important.